Great. Okay, everyone. Hi, welcome to the second VanBug of the season. Today, I would like you to all at least silence your phones. Sorry about that. I forgot this slide was here. All right, if everyone could please silence their phones throughout the duration of the talk, that would be great. And just so you know, this event is being recorded. So please, if you have any questions, please contact William Shaw over there. Otherwise, without further ado, we'll get to our schedule for today. We'll have a few announcements followed by our trainee talk and then our featured talk with Dr. Jessica Dennis. And then we'll have at 7:15 some time for networking, food and drinks, and then we'll be out of here by 9. I'd like to thank the VanBug organizing committee for getting us all here today and getting everything set up. And also upcoming in November, we will have Emma Griffiths talking about her work. She's from the Center for Infectious Diseases and based at SSU. And our student speaker will be Faiza Kashavars. And for anyone who is interested in VanBug going forward for the next year, please go to our website at vanbug.org to suggest a speaker. We take in all suggestions and we vote on them at the end of the year. We are sponsored by the Bioinformatics training program at British Columbia, UBC and SFU. We're also sponsored by Genome BC, and we're sponsored by bioinformatics.ca. Imagia Connexia Health, Langara College, and we're also sponsored by Stem Cell Technology, and last but not least, St. Paul's Hospital, where we're speaking from today. So VanBug is also affiliated with 3 different groups. We're affiliated with the International Society for Computational Biology, ISCB, and also our sister bug groups in Montreal and Toronto, which are MonBug and TorBug, respectively. As I said, our featured talk today is Dr. Jessica Dennis, and I'll introduce her after our student talk. And our training talk today is by Field Demayakiak. Sorry about that, I didn't ask you first. And then his talk will be on evaluating the adequacy of phylogenetic data, phylogenetic comparative methods for gene expression data. So without further ado, welcome Field.
Hi, everyone. So today I'll be presenting my work on evaluating the adequacy of widely used phylogenetic models for gene expression evolution. My name is Fiel Demiakiak from currently staying at the Pavlidis lab, but my main supervisor is Dr. Matt Pennell. So one of the reasons why we want to study gene expression when it comes to evolution is that comparing gene expression between species can give us insight into the evolution of diverse phenotypes. So for example, in Chen et al. 2021, they used differences in gene expression to predict the susceptibility of different organisms to SARS coronavirus 2. Specifically, they used the expression of the ACE2 receptor. And in Stern and Crandall in 2018, they compared gene expression values between species to identify the evolutionary processes that cause the repeated loss of vision in cave-dwelling organisms. And they found that it was due to the relaxation of selection pressures on the gene expression related to their blindness. Now, a set of tools that can be used to make hypotheses about evolution by comparing traits between organisms are phylogenetic comparative methods. Now, these methods are powerful because they can account for the underlying relationship between different species given a phylogenetic tree. So historically, these methods have been used with more easily observable traits such as wing size or say tooth length. But recently, these PCMs have been used to study gene expression, like in that Stern and Crandall paper I just mentioned, or in Chen et al. 2019, where they showed that gene expression in select mammalian tissues can be modeled by what is known as an Ornstein-Uhlenbeck process, which is one of the PCMs I will be talking about later. Now, this is great, but this is also a new context for PCMs, and it represents a potential problem because phylogenetic comparative methods have not yet been assessed for this new context. So what my work aims to do is to do this assessment, and specifically to assess how well commonly used phylogenetic models work for gene expression datasets specifically. So what are these models? Well, one of the models I'll be testing today, or in my work, explaining today, is the Brownian motion model. Now, this model is a random drift of a character trait along a phylogeny. So the only parameter in this model is the evolutionary rate, so how fast the trait changes over time. And because the only parameter in this model is that rate, this model is often used to as an analog for genetic drift. The next model, and the one I've mentioned previously, is the Ornstein-Uhlenbeck one. This model uses the Brownian motion model as a basis, but adds two parameters that make it useful for modeling stabilizing selection. These parameters being an evolutionary optima, so the optimal value for some character trait, and a selective pressure, which can be considered, you can think of it as a rubber band, keeping the trait towards that selective optima. The last of these models is the early burst model, which I will not be going super into depth into today, but it is often used to model adaptive radiations. So in the field, when it comes to model assessment, typically the approach is to find relative model fit. Specifically, we tend to use the AIC, which is a metric that balances accuracy of a model with a penalty term for adding free parameters. In this way, you can account for the problem of overfitting. Now, while this is great to use in a relative sense, if given a set of models, none of them adequately explain the variation of the data, a relative fit test will still select one. Now, when we are evaluating a set of models in a new context, which of these models is the best is not the question we should be asking. Rather, we should be asking the question, does the model actually account for the variation of the data? So in other words, we need to measure phylogenetic models in an absolute sense. So in my work, I use the Arbutus tool from Peddle et al. 2015, which in short provides a set of 5 objective test statistics that measure model performance in a phylogenetic context. So it operates on a relatively simple concept where if a model is good, one should be able to use the parameters of that model to re-simulate the data that you originally gave it. This is known as parametric bootstrapping. So given a set of simulated data, what Arbutus does is it compares the observed data to the distribution of the simulated data. And if it lies within that distribution, the model is good. And this process, because it is kind of analogous to a t-test, generates a set of p-values. But in this case, instead of wanting our p-value to be below that threshold of 0.05, we actually want it to be above that threshold. So in other words, a model is adequate if it can account for the variations seen in the data. And I will be identifying in which ways models succeed and in which ways they may need improvement using these 5 objective test statistics. So to understand my overall analysis process, let's work through an example data set. I got compressed a bit. So this data set is comprised of the transcriptomes of 21 different species for 6 organs amalgamated from 182 different data sets. So my process is relatively simple. Given a set of gene expression data and a phylogeny, for each gene, I fit the 3 models that I mentioned earlier, Ornstein-Uhlenbeck, early burst, and Brownian motion, and selected in a relative fit test which of those 3 models was the best fit, again, in a relative sense. And given that best fit model, I then assessed the absolute adequacy of that best fit model for each gene. So what does the data look like? Well, the best fit results are seen as the left. As you can see, for this specific data set, most of the genes were best fit by an Ornstein-Uhlenbeck process. Not really a surprise, considering it has been shown previously to be good for this kind of data. And on the right, we get those p-values I mentioned before. So black bars represent a threshold of 0.05, and each column is a different one of those 5 test statistics or 5 objective test statistics I mentioned earlier. As you can see, of the 5 test statistics, they tend to not be displaying any prominent tails towards the 0.05 end, and the values are distributed relatively evenly. So to me, this suggests that for this data set, which is primarily best fit to an Ornstein-Uhlenbeck process, that the best fit model is relatively adequate for the data, and that it can explain the variation in the data, mostly. I carried this analysis out on 9 different data sets, which range in number of genes, number of species, types of taxa, and organ type. And overall, this is what I found. So firstly, that model adequacy is, firstly, that on the right, we see that 61% of genes were fully adequate. Overall, they were not inadequate in any of the test statistics, which is a good sign because it means across the data sets, our best fit models tend to be doing at least okay. But on the left, we see a slightly different picture. There was tails near the lower end of the p-value distribution are now present. So as we see across data sets, across gene expression data sets, best fit models tend to be inadequate in 2 specific ways. I won't be going into a lot of detail about what they are, but they are related to heterogeneity and evolutionary rate, which means there are issues in our simple models that cannot explain multiple rate parameters. So for example, if the true generating process, if in reality gene expression evolved at different speeds across different taxa, which could be the truth, then our models are not accounting for that at all. So in conclusion, measures of relative fit pick the best model out of a set of models, whereas measures of absolute fit, again, adequacy, as I mentioned before, determine how well a model captures the variation seen in the data. Second, Ornstein-Uhlenbeck models are typically the best fit models and have good adequacy for many gene expression data sets. And again, overall, 61% of genes are fully adequate when the best fit model is used, which again, tends to be Ornstein-Uhlenbeck. And lastly, when models are inadequate, they tend to be so in 2 very specific ways, which are both related to heterogeneity and evolutionary rate. Thank you, and accepting any questions.
All right, thank you, Phil. So we have time for 1 or 2 questions. Today we worked out the mic system here. So in order to ensure everyone online also hears your question, please touch the little plastic ring on the mic in front of you and speak into the mic, and it will pick you up. So any questions for Phil? Yep, go ahead.
Between the species you're considering.
Like orthologous genes?
Yeah, how are you stringently like allowing one-to-one comparisons essentially?
Oh yeah, I see what you're saying. So that's actually one of the things I filtered for when picking data sets. I made sure that they had one-to-one orthologs. I go into more detail in the rest of my analysis comparing gene family relationships, species family relationships, and it can get pretty complicated. Most genes don't have one-to-one orthologs. Okay, maybe not most, but a lot of genes don't. So.
Would you imagine that if you were to pick like a family set that might have been excluded, that could change some of your observations, like something that was originally paired by your processing might actually be closer to another family in another species, or sorry, within the same family but a different gene that was considered a one-to-one?
Yeah, it's definitely possible that they will behave differently with models. More complicated models would probably be better for, especially in instances where you don't have one-to-one relationships between genes and species. This disconnect between the topology of a gene family tree and a species family tree is definitely a possible confound. And well, I know it is because I actually checked it. Oops. Here. Okay, I'm just gonna leave that. Yeah, so this on the top row is the I ran adequacy tests on gene families for the same data set, and then I used the species phylogenetic relationship on the bottom. And yeah, it does matter. Yeah, quite a lot actually.
Okay, with that, Saul, thank you again for his wonderful talk. Yeah, I got this. Yeah, go ahead. And I'm just going to introduce our next speaker here. Great. So Dr. Jessica Dennis is an assistant professor in the Department of Medical Genetics at UBC, and she's an investigator at BC Children's Hospital Research Institute. So by training, Jess is a genetic epidemiologist, and she applies methods to emerging big data resources in population health. So essentially, she aims to understand how genetic, epigenetic, and environmental differences between people contribute to variation in the susceptibility to different diseases, and with the end goal of aiding treatment and recovery. So a primary goal of her research is to reduce suffering associated with psychiatric and neurological disorders, which together account for more years lost, disability, and death than either cancer or cardiovascular diseases. And she conducts all of her studies in large population data sets with a major interest in electronic health records and biobanks. She completed her postdoctoral training at the Vanderbilt Genetics Institute at the Vanderbilt University Medical Center in Tennessee. She holds a PhD in epidemiology from the University of Toronto, where she was fellow in the interdisciplinary CIHR-STAGE program. And she holds a master's in epidemiology from the University of Ottawa and a bachelor's of science in biology, minor in genetics from the University of Guelph. Today, Dr. Dennis will be talking about genetic epidemiology in the era of big data, biobanks, and omic technologies. So without further ado, I will pass off to Jess, and she will talk. Okay.
Thank you, Will. Good. Okay. So, hello again. For those of you who were here earlier, hello again. For those of you who are new to the room, welcome, hello. And as you heard from Will's introduction, I consider myself to be a card-carrying genetic epidemiologist, which those of you who are in the room earlier heard me talk a little bit about. And for those of you who might be a little less familiar with what it means to be a genetic epidemiologist, don't worry, I'm going to cover that as part of this presentation. But before we get into that, I wanted to actually start by admitting to you that I wasn't always as confident or as proud to be a genetic epidemiologist as I am now. And that is despite doing a PhD and a master's degree in epidemiology and being part of a dedicated training program in genetic epidemiology as part of my PhD. So I had all of this training. But the challenge was when I came out of my PhD, there were all of these other graduates who had really cool titles, things like bioinformaticians, human geneticists, computational biologists. And I didn't really know what I wanted to be because all of these things sounded so cool. So when I was in my postdoc, I tried on all of these different titles when speakers would come in and we had to give our little elevator pitch and describe who we were. Sometimes I would describe myself as a computational biologist. I didn't really know what it what I wanted to do and what I was trying to be. And when I stopped panicking and I looked at this richness of data around me, these big data sets, the biobanks, these omic technologies, I realized that it really was the tools of genetic epidemiology that I wanted to use to understand the biology that was hidden in all of these data sets. And so that's what I want to highlight to you today and showcase to you today is how we can use genetic epidemiology and how my lab is using genetic epidemiology to understand the biology that underlies disease risk. And so somebody asked me in the Q&A session before, what is my motivation? So I want to stop here and thank the organizers for having me, as well as thank you guys for listening, because giving a talk like this gives me the opportunity to take a step back from my research, think about why I'm doing what I'm doing. And for me, one of the big motivations is my children. So I have 4 kids. This is them last year. The 2 little ones are twins. The twins are now 2. And I was asked this past Mother's Day by BC Children's Hospital, where I work. The communications team asked me to provide a quote about what being a mother, how being a mother influences what I do in the research space. And I said this to them. I said, my children are ridiculously curious, which inspires me to ask lots of questions in my research. Last week, my 5-year-old asked me, do fish toot? And I have asked this of my graduate students several times. Nobody has yet to give me an answer, so I still put it out to any of you to answer to me if fish toot. I haven't looked it up yet. But given that many of you are at the beginning of your graduate school journey, I think that pausing every so often and reflecting on why are you heading down this path? Why are you doing what you're doing? What is that motivation? I think it's important because it's not always going to be easy, and connecting regularly with what it is that motivates you can be really helpful when the going gets tough. Now, with that aside, I want to talk to you a little bit more, like I promised, about genetic epidemiology in the era of big data, biobanks, and high throughput omic technologies. And to have this discussion, we're going to use the following outline. So first, we're going to talk about the objectives and study designs used in genetic epidemiology studies and give you a real flavor for what I consider, for what I think genetic epidemiology is. And then the second half of the talk is going to be talking about post-GWAS analyses, because it's a bit of a spoiler. Section 1 is really about GWAS, or genome-wide association studies. In Section 2, we're going to talk about what we do after you've conducted that GWAS. A silver lining of the pandemic, I think, is that epidemiology became more of a household name term. So before the pandemic, when I would describe to people that I had a PhD in epidemiology, I would often get the question or the response, so you work with skin, right? Now, at least post-pandemic, people understand that an epidemiologist is the person who's doing the science that drives the public health policy that kept us all safe. In actual fact, though, what we do in epidemiology is, probably you might have guessed, is we sit at a computer screen, we analyze data. And more formally, epidemiology is the study of how often diseases occur in different groups of people and why. And perhaps the downside, one of the downsides of the pandemic was that epidemiology was very strongly associated with infectious disease. And I want to emphasize to you that epidemiology is not restricted to infectious disease. In fact, most of what my lab studies is chronic diseases, chronic complex diseases, brain-related traits and conditions, as I said to some of you before this talk. So genetic epidemiology, moving from epidemiology to genetic epidemiology, is the study of how genetic factors influence human traits and diseases in families and in populations. Genetic epidemiology is built on a tradition of rare single diseases in family-based studies. So on the right-hand side of the slide there, you can see an example of a single gene disease. So things like something like sickle cell anemia, where it's really just about a single place in the genome where there's a mutation that confers disease risk. So that's where genetic epidemiology really started, studying families with these rare genetic conditions. Now, a lot of what we study in genetic epidemiology is these complex diseases, like you can see on the left-hand side of that figure. A condition like ADHD, attention deficit hyperactivity disorder, where there's many genetic variants, not just a single genetic variant, but many genetic variants acting in combination and in interaction with the environment to confer disease risk. So much of what we study now is complex or multifactorial conditions and diseases and traits in population-based studies. And a lot of the advances in genetic epidemiology have been driven by this explosion of biomedical data over the last 15 years. And a lot of this biomedical data is captured in biobanks. And depending on your space, biobanks mean different things to different people. So when I started at BC Children's Hospital, everybody said, oh, BC Children's Hospital has a biobank. There are about 300, 500 kids with biological samples and some phenotype data. But the biobanks that I work with are really, really large-scale biobanks of hundreds of thousands of people. What I mean by a biobank is a repository of biospecimens like blood linked to phenotypic data. So example would be data from questionnaires or electronic health records. And why I think biobanks are so exciting is that they enable this data-driven approach to quantifying, characterizing, and understanding the relationships between biospecimens and the phenome. And sorry, there's a little glitch there. The biobanks that you might have heard of, and as I said, the biobanks that get me really excited are those large-scale biobanks that have hundreds of thousands of participants. So, for example, the UK Biobank or 23andMe, for example. And just by show of hands, who has heard of the UK Biobank or even worked with UK Biobank data? Okay, so about 2/3 of people. Out of curiosity, who has done a 23andMe genetic test? Who's done 23andMe on themselves? Will has. Okay. 23andMe, who knows about 23andMe? Okay, so everybody knows about 23andMe. I have done a whole separate lecture in the past about 23andMe and what it means, which I won't get into at this point, but happy to talk about it over pizza and soda afterwards if people are interested. So one of the main tools that is used in biobanks is a genome-wide association study. And again, I'm going to ask you to participate here. So maybe just by how high you raise your hand, how comfortable are you with genome-wide association studies? So this is like, yeah, I know GWAS. Okay, so we've got some variation here. It's good to know my audience. I will take a little bit of time here describing what a GWAS actually entails. So in a GWAS, you start with data collection. That panel A is about collecting data on a phenotype, some sort of trait or condition. Let's say it's ADHD. You start by collecting that phenotypic data on your population. Panel B, you genotype them. You do some elements of quality control, which, yes, Cindy, are very important. They're key part of this step. That was a question earlier. You do some imputation and then association testing. So that panel E is the results, the Manhattan plot that you get from a GWAS, because it looks like the skyline of Manhattan, where each dot is an association between a genetic variant in the genome, and the y-axis is the strength of association between that genetic variant and the phenotype that you've collected information on in panel A. Because you're doing so many statistical tests, you have to have some stringent multiple testing and meta-analysis and replication standards, which is what you see in panels F and G. And this is a really, really powerful tool. And I love showing this figure because it shows the growth of GWAS between when GWAS were first proposed in 2007, right up until, I think the final slide ends in 2019. And what this figure is showing you is the number of associations this tool has identified. So each of those vertical bars is a chromosome. Each of those dots is an association between a genetic variant on that chromosome and thousands of different conditions and traits. If you can measure the phenotype, you can do a GWAS on it. And there have been some really surprising GWAS, like a GWAS of the number of children that you have, or a GWAS of how much coffee you drink. It's almost like if you can measure it, you can GWAS it. And one of the coolest GWAS that I have been involved in is a GWAS of loneliness. Now, loneliness, I think, is a very interesting trait because it's a subjective feeling. So loneliness is a feeling of discontent with your social connections. It's distinct from objectively measured social isolation. So it's how somebody feels about their social relationships. And 22% of adults are chronically lonely. This was even before the pandemic. I know loneliness got a lot more play, a lot more conversation during the pandemic, but these were statistics from before the pandemic. And this is such a big problem that there were, the UK had even appointed a minister for loneliness to recognize how big of a societal problem this is. Because as you can see from this slide, that people who have a feeling of loneliness are at increased risk of early mortality. And I also want to contrast loneliness with a feeling of depression or major depressive disorder. Major depressive disorder is a feeling of discontent with life in general. It has a 17% lifetime prevalence, and it has a much clearer sex difference compared to the feeling of loneliness, and also a stronger risk of early mortality. Loneliness can lead to depression, but they're only about 40% comorbid, which means that they're found together in the same individuals about 40% of the time. So they're overlapping, but also distinct. So like I said, we did a GWAS of loneliness using some of those big biobank data sets that I introduced a few slides ago, namely the UK Biobank and data from 23andMe. So this, as well as a couple of other smaller data sets. So we had a total of 280,000 participants in our GWAS of loneliness, and we found 16 independent and significant loci, as you can see in this Manhattan plot here. Now what? The panel that I had omitted when I was first introducing GWAS is this final panel, which is on post-GWAS analysis. So actually doing a GWAS is pretty straightforward. Yes, you need to do rigorous QC. There are some hiccups you can run into along the way, but doing a GWAS is pretty straightforward nowadays. The real fun and challenge begins in this post-GWAS analysis phase, and it's also where biobanks shine. So now we're going to move into the second part of the talk, where I'm going to talk about, after you've done the GWAS, what do we do? The first section, part A, 2A, is about polygenic scores. Again, let's stretch our arms by virtue of how high you raise your hand. Polygenic scores, yeah, kind of familiar with, or I really know polygenic scores. Okay, so again, a good mix here. So we had our 16 independent and significant loci from the GWAS. And we wanted to understand whether the feelings of loneliness that were associated with a 38% increased risk of early mortality, if you recall from that first slide where I introduced loneliness, was it because of genetic pleiotropy? Is it that the genetic variants that increase your risk for feeling lonely, are they also the genetic variants that increase your risk of dying early? Or is it due to comorbidity? Is it that when you feel lonely, then you're more likely to engage in behaviors that increase your risk of early mortality? Something like smoking. If you're feeling lonely, do you engage in smoking behavior that increases your risk of early mortality? So those were kind of the two things we were trying to tease apart here. And to tease this apart, again, we turn to a biobank. And this is where I did my postdoc at Vanderbilt in Nashville, Tennessee. And I described it as a sandbox for a genetic epidemiologist, like really this amazing resource, this electronic health record linked biobank. And that's because everything that happened on the clinical side, notes that were taken in a clinic, medications that were dispensed, clinical lab values that were measured, patient demographics, registry data, vital statistics, any billing codes, as well as outpatient visits, that was all de-identified and pushed through to this research data warehouse. And we had 2.8 million patient electronic health records that covered 10 years of patient encounters on average. So from an epidemiological standpoint, that's huge. And then for 10% of people, we also had a linked DNA sample. So that from a genetic epidemiological perspective, that is also this amazing resource. So now we come to those polygenic scores. A polygenic score is an allelic sum weighted by the effect estimate from your GWAS. To walk you through that a little bit more visually and formulaically, on the left-hand side, you have those results from the loneliness GWAS. So each line in that database is the association between a genetic variant and GWAS and loneliness. So the risk of loneliness conferred by that genetic variant. So with those GWAS results, you can then go into your biobank like that Vanderbilt Biobank, and for each patient for whom you have genetic data, you can calculate their polygenic score for loneliness. And that looks like this. For patient 1, you would calculate their polygenic score as the sum of the allelic effect times the number of copies of that allele that this patient has. So for patient 1, they have 0 copies of SNP1, so 0 times the allelic effect of SNP1, plus they have 2 copies of SNP2, so 2 times the allelic effect of SNP2, and so on for all the SNPs, all the genetic variants that you want to include in that polygenic score. So big picture takeaways, what I would like you to take away about a polygenic score is that they quantify an individual's polygenic risk for a trait, and they can be calculated on anyone with genotype data. So you could calculate my polygenic score for loneliness. We could calculate Will's polygenic score for loneliness if I had his genotype data. We could calculate this Will's polygenic score because 23andMe has his genetic data. And what we can do really cool things with those polygenic scores. One of those things we can do is a phenome-wide association study. Phenome-wide association study, I'm going to contrast it to a genome-wide association study here. So on the left-hand side in the blue, we've got a GWAS. And in a GWAS, you start with the phenotype and you look at the relationship between that phenotype and all the genetic variants in the genome. In a phenome-wide association study, you flipped the two. So you start with genotype, whether it's a single genetic variant or a polygenic risk score, you start with that genetic variant, and then you look at the relationship between that genetic variant and all in your entire phenome. And you can imagine somebody's electronic health record is super valuable for that because you know all the diagnoses, all the medications, all the lab values that have been measured on that person. And using this PheWAS approach, we can distinguish comorbidity from genetic pleiotropy, which if you recall, that's the main thing that we were asking when we had these GWAS results of loneliness. So this is what a PheWAS of the polygenic score for loneliness looks like in the Vanderbilt University Medical Center Biobank. So this was a PheWAS of 18,000 participants, and we analyzed 897 different Phe codes. So those would be medical diagnoses in these patients' medical records. And the plot, the way you would read the plot is kind of like you read a GWAS, but on the x-axis here, instead of genetic variants, these are those 897 different Phe codes or medical diagnoses. And on the y-axis, this is the strength of association between the polygenic score for loneliness and those different medical diagnoses or Phe codes. And you can see some Phe codes that pop up that you would expect. We know that loneliness and major depressive disorder are comorbid, so we do see this association between a polygenic score for loneliness and depression, tobacco use disorder. We also, however, see this really strong signal in the red, and these are cardiovascular disease-related phenotypes. You've heard of the expression dying of a broken heart. This is perhaps telling us something about what is going on in your genetic predisposition to loneliness and that risk of early mortality? Is it related to cardiovascular disease? Is it that old adage, dying of a broken heart? Are the data telling us something about genetic pleiotropy, or is it really just comorbidity? So to get at that, the next thing we tried to do was we tried to kill that signal. We tried to kill the association between the genetic risk for loneliness and your risk of coronary artery disease. And I think if there's one thing that you take away, try to kill your signal. When you're doing your research, if you can't kill whatever signal that you found, then that's a really good thing. But if you can kill the signal, it's probably a false positive. So we tried to kill the signal. If you follow along, I'm going to show you all the different ways that we tried to kill the signal. This is a forest plot. So to orient you here, we're going to track this dot as we add different covariates to the model. And if it crosses this odds ratio value of 1, that means that we've killed the signal. And you can see when we added in different conventional heart disease risk factors, yes, depression and heart disease are often comorbid, so that's a conventional risk factor, but things that you might be more familiar with, like BMI, hypertension, smoking, highest level of education, type 2 diabetes, a polygenic score for coronary artery disease, blood levels of HDL cholesterol, LDL, and triglycerides, we couldn't kill the signal, which was telling us that there was some genetic pleiotropy happening, that there was something about the genetic risk for feeling lonely that also conferred risk for heart disease or coronary artery disease. And then we did a little bit of multivariate modeling to try to tease apart, was it something about loneliness? Is it depression? What's going on? And so here you've got, again, this forest plot where we're looking to see if the odds ratio crosses 1. And on the left-hand side, this is a polygenic score for depression when we've taken out the component that's comorbid with loneliness. And we're not seeing an association anymore. Here, we actually have killed the signal. Conversely, when we look at loneliness and we pull out the part that's comorbid with depression, we still do see the signal. And we actually see a stronger effect in females compared to males, which is telling us that the genetic factors that are unique to loneliness and not shared with major depressive disorder, those genetic factors are increasing coronary artery disease risk. But those that are unique to major depressive disorder are not. And bringing this, I think, kind of full circle and maybe even tying in with the first talk about evolution, I think it's really cool to think of loneliness through an evolutionary lens. So humans are among the many social species who have evolved to depend on each other. And loneliness serves as a painful alert that you're on the social perimeter. When I started this project, I thought, why would there be any genetic variants that predispose you to feeling lonely? And it makes sense when you think about it from this evolutionary perspective. And there are known physiological effects of loneliness. They trigger depressive symptoms. Loneliness disrupts your sleep, raises blood pressure, and activates the HPA axis. And so these are all biological mechanisms that we know are also associated with coronary artery disease. So we think that there's something that we learned about biology by doing all of these things. Before we move on from this section, I think it's really important to acknowledge that there are gaps in terms of genetic ancestry that we analyze in a lot of our biobanks, a lot of our GWAS. Most GWAS are on European-based populations. So this is a figure where you can see that the global population here, in terms of ancestral diversity, and then the individuals included in GWAS on this axis. And we've got this gross overrepresentation of European ancestry participants that is completely disproportionate to their global population. And another major gap is that differences between sexes are often overlooked. And these are gaps that two students in my lab have been looking at in their thesis. Paola's at the back. She will happily talk to you about this over pizza and soda. But again, these are gaps across all of science. And so when you go into your research projects, I encourage you to think about these gaps as well, because it's not unique to biobanks. It's not unique to GWAS. So finally, the very last section, 2B, is talking about the functional interpretation of GWAS findings. And some of you might be very well familiar with this statistic, but only 2% of the genome is actually protein coding. The majority of the genome is involved in gene regulation. So if you think in a GWAS, we're testing millions of genetic variants across the genome, even just probabilistically, most of the genetic variants that we find in a GWAS are going to be outside of those protein coding regions. And that is objectively what we find too. 90% of the genetic variants that you find in a GWAS are outside of protein coding regions, which means that there's this big challenge facing us now of how do we interpret the functional significance of these genetic variants in terms of the cell types they're active in, the tissues, the developmental stages in which they're active, as well as the environments that are relevant to these genetic variants. So this is a major thrust of the research that we do in the lab. And I know there's a lot on this slide, but I think it captures just the complexity and the challenges ahead of us so well, because in the gray, you can see what we're testing in a GWAS. So in a GWAS, we're testing the relationship between genetic variation and your phenome. But there's all of these different omics in between your genetic variation and your phenome. There's the epigenome, there's the transcriptome, there's the proteome, there's the metabolome, and all of these omes are mechanisms by which your genetic variants could be affecting disease risk. And until we know those different biological mechanisms, it's really hard to actually understand what we're finding in a GWAS. And these different omics are also specific to cell types, tissues, developmental stages, and environments. And so the tissue, the cell type that I am interested in these days is the placenta. And this might not be surprising to you, knowing that I grew 4 placentas inside of me over the last 8 years. They are just phenomenal organs. If you stop and think about it, they are the organ or the tissue that supports our earliest development. They are our environment for the first 9 months of our life. And they are so often ignored. I mean, any of you who have delivered a placenta, you know that it just goes in the medical waste. So few people are studying this tissue. But there's so much more epidemiological and genetic evidence.
Code blue, Providence 8A. Code blue, Providence 8 alpha. Code blue, Providence 8 alpha.
So there's growing epidemiological and genetic evidence that healthy life trajectories start at conception. When we look at the genetic variants that we find in a GWAS, they're often active in fetal brain. It's not when the disease is manifesting, it's what's happening prenatally. So I'm really interested in the placenta and how the genetic variants that we identified in a GWAS might be affecting those different omics data types in the placenta. And this is a project that Will, here in the front row, has tackled in my lab. And what he's done is he's looked specifically at methylation quantitative trait loci. So these are genetic variants that are associated with levels of methylation in the placenta. And he's looked at it in males and females. And then he looked to see how these methylation quantitative trait loci were enriched in those GWAS, those many different GWAS data sets that we have out there now. And in this plot, so enrichment above 1, or values above 1 is showing significant enrichment, and it might be a little bit small, but those tiny little red, those little hashtags are enrichment results that survive multiple testing correction. So when we started this project, I was really excited that we might be able to find something about the placenta and early life in neuropsychiatric disorders, because like I said to you all earlier, I'm interested in brain-related traits, but most of what we found actually was immune-related traits and growth-related traits. So it's telling us that the placenta is having an important role in the placenta is having an important role in contributing to risk of these immune-related traits, things like type 1 diabetes, child onset asthma, and hay fever or eczema. So these are allergic type conditions. And we're also seeing that when we look at it through a sex stratified lens, so we look at those MQTLs that are specific to either females or males, we are seeing different things than when we look cross-sex. So again, if I haven't convinced you yet that sex differences are important, I hope Will's project helps to convince you of that. After Will looked at enrichment, the next thing he did was look at colocalization. So colocalization using a statistical method that looks to see if the same genetic variant is underlying both the GWAS results and the MQTL signals. So you can see it visually here on the left-hand side where we've got a GWAS of child onset asthma. There's my cursor. So GWAS, here's like an associated region of child onset asthma. And this is also where we're finding a genetic variant that is associated with placental MQTL in both sexes, and in this case, in females and males stratified as well. And rolling that up across multiple different traits, this is the plot that you see on the right-hand side, where these are the number of different colocalized CPG sites. So these are sites that have a genetic association as well as an association with DNA methylation in the placenta. So again, telling us that the placental MQTL are colocalizing with SNPs that are identified in the GWAS, and the placenta is contributing to the risk of these childhood onset traits. In summary, so the take-home messages I have for you all this evening: big data, biobanks, and high throughput omic technologies make it a great time to be a genetic epidemiologist. I am proud and confident to be a genetic epidemiologist. Differences across race, ethnicity, and ancestry, and between biological sexes are an important next frontier across all of science. And the real fun and challenges begin after your GWAS results are collected. And finally, to come back to that motivation question, I encourage you to connect regularly with whatever motivates you as you carve a path through your research career. Finally, I'd like to thank my lab, many of whom are here today, and colleagues here at UBC and at Vanderbilt. And thank you all for listening.
Okay, so we have another 15 minutes for questioning. If Dr. Dennis would like to go more than 15 minutes, you have time for that as well. So like before, just touch the plastic ring in front of you, raise your hand, we'll call on you, and then once it glows, you can go ahead and ask your question.
Okay, right in the middle in the green, please.
Hi, thanks so much for that talk. I was just wondering, when you were talking about the biobank that you were using earlier for the loneliness phenome analysis.
Yeah.
You mentioned that 10% of the patients that you had DNA samples, and I was wondering if you guys took into consideration of why or reasons why those patients would have those DNA samples. So for instance, if there are patients, you can imagine, for instance, like patients that are, maybe dealing with multiple severe illnesses, they're more likely to have that DNA information as opposed to just your average individual or something like that.
Yeah, no, great question. And there certainly are some there's ascertainment biases in any data set that you work with. The very cool thing about the Vanderbilt Biobank was, for a number of years, as it started, it was default opt-in. So actually all patients, when they came, we weren't even consented. You just, by default, by seeking care there, you were opted into the biobank. And then the NIH changed rules, and it became that you had to consent in. But it turns out that there wasn't a huge drop off when people had to actively consent. So in terms of self-selecting into the biobank, I don't think that there was too much bias there. So the participants in the biobank aren't too ascertained. There was some ascertainment in terms of who was genotyped in the end, because genotyping is expensive. You're not going to genotype everybody all at once. And there was an enrichment of patients with cardiovascular diseases in some of the early data analysis. What I showed you today, however, patients were selected for genotyping if they had long medical records. So yes, there is a selection for people who have perhaps more complex care. I think it was like 3 different visits over a course of 5 years. But that's a pretty low threshold. I visit the doctor 3 times in 5 years, and I think it's a reasonable threshold. So I don't think there's too much ascertainment there. And actually, a result I didn't show also was that we replicated in another very well recognized cohort study called the Framingham Heart Study, for those of you who are in that epidemiological space. we replicated in a population-based community cohort study as well. So I think that's a great question about ascertainment and who's in your biobank. It's something you definitely have to think about, but we're a little bit more confident that these results are robust. We didn't kill the signal in the Framingham data set.
We have a couple online questions.
Sure.
From Brooks Perkins-Show. Is the substantial increase in mortality for major depressive disorder due to suicide? Or what is that mortality?
So the mortality associated with major depressive disorder, was that the question?
Yeah.
Yeah. It's not actually. I mean, certainly there's an elevated risk of suicide in people with serious mental illness, things like schizophrenia and major depressive disorder, treatment-resistant major depressive disorder. But as far as I can recall, the big sort of population-based epidemiological studies that have looked at cause of death, actually the primary cause of death in people with serious mental illness is cardiovascular disease. There's more and more recognition that there's this brain-body connection, that biobanks really are leading the fray in finding these associations, because a lot of the early studies that have, like the Framingham study, was just developed to look at cardiovascular disease and wasn't necessarily well equipped to look at the relationship between cardiovascular disease and mental health. So the biobanks that collect this data agnostically without a hypothesis in mind to begin with can be really useful in breaking down those boundaries.
We can take another question.
Eric.
Thank you for the talk. I have a question, which is, how do you incorporate in the context of a GWAS the role of the environment? So I'm just thinking, like, for example, like maybe if you do a GWAS, a SNP is not going to be associated because it needs a specific environment that kind of like activates it.
Mm-hmm.
So like, is there any like methodology that kind of incorporates the environment into the tests?
Very crudely. I mean, you can stratify by environment. So there have been some GWAS where we've looked at genetic variants that are associated with depression in people who were exposed to childhood trauma versus people who are not exposed to childhood trauma. So you can do, certain very basic gene environment interactions that way. But to do a true genome-wide gene environment interaction is a very heavy multiple testing burden and has computational challenges. As big data sets like the UK Biobank become more widely used, there are new methods to look for gene environment interactions, and especially looking at the entire exposome, so every single environmental variable. It's part of this big omics space. So there are methods that are tackling that, but you do need very big data sets, like UK Biobank scale data sets, not child.
So we have another question online from Jeremy Fan. So can polygenic risk scores be extended to structural variants or any other type of genetic variation?
Yes. So there are a few methods that combine polygenic scores and rare variants. That is a newer space I'm less familiar with, but I know that there are some approaches that recognize that, common variants, as you would capture in a polygenic score, are only part of the picture. I think there was a question at the back that keeps coming up.
Hi, thanks. I actually have two questions about this.
Sure.
The first one is you've alluded so much to the fact that you should look at the differences between sex and race. And I was wondering, with the link between loneliness and those SNPs, were there any sex or race specific SNPs that weren't, were there differences between those groups?
So sex, yes, and we were equipped to look at sex differences in that polygenic score. I don't think we actually sex disaggregated the GWAS results, which we should have, and we were trying to get the data from 23andMe to do that post hoc, but 23andMe is a bit of a black box to work with. This is getting into a little bit of what I talk about with the students as part of that lecture, but I think 23andMe does some really good things and has fueled a lot of science. They have this very big data resource and they do collaborate with scientists, but they're also a company. Right. So getting the sex disaggregated results from them, we're still working on it. In terms of genetic ancestry, we did not. I mean, that was a GWAS of 2019, and we should have. And 23andMe, one of the good things that they're doing is they are prioritizing project proposals that are interested in diverse genetic ancestries. So there's a push. We did not look at it. The Vanderbilt dataset now, I think about 20% of the genotype participants are of African-American genetic ancestry. Those weren't the participants that we analyzed in our dataset, but it's something that we're trying to move forward on.
I see. It's really cool. Thanks. And then my second question was, I was wondering, is the link between mental health and a physiological thing like cardiovascular disease is fascinating. And I was wondering if in the data set it's possible to see whether there are more cases of loneliness without cardiovascular symptoms, or if there are more cases of cardiovascular symptoms without loneliness, or vice versa, like the fidelity of that relationship.
Yeah, so in the data set, we didn't actually have a measure of loneliness in the participants in the Vanderbilt Biobank. We do have measures of depression though, and it's an ongoing project. A PhD student in the lab I used to work in, an ongoing project is to look at those temporal relationships, look at that overlap, both from an epidemiological perspective as well as genetically. So how strongly genetically correlated are these different conditions? Because the temporal relationship is also very complex, and it's actually quite cyclical. That it's not a linear thing. They both it all works affecting one another.
It's really interesting.
I think Andrew first.
Yeah, I was just wondering, so for loneliness and for major depressive disorder, like women have a much higher rate, and I think generally for most mood disorders, women would have a higher rate. Is that right?
Yes.
But men have a much higher burden of suicide. So I was wondering what your thoughts are on that.
So the difference in reconciling the difference that mood disorders like major depressive disorder are more prevalent in women than men, but suicide being more prevalent in men than in women or in males. I don't have a good answer for that. I think that's an important question. I'd be curious when you break down, like depression is such a heterogeneous condition. I would be curious at the sex differences when you look at treatment-resistant major depressive disorder or severe recurrent major depressive disorder, and I'm actually not sure what the sex differences are there, if it's more prevalent in males than it is in females. I don't have a satisfying answer. I would probably bring in elements of social support and how likely women are to seek support compared to males, females compared to males, I should say. Yeah, sorry, that's unsatisfying,
No,
but an important question.
yeah, so a lot of things going into it. So.
Yeah. I think there was a question there first.
Yeah, I wanted to ask something. First of all, thank you very much for the talk. I was thinking more on the side of an epidemiologist obviously has to do a lot of meetings. I think that was something that is always missing. Like you have to connect with a lot of stakeholders, and from your data and from many other works that we have seen recently, we noticed that the amount of linked data, like especially like clinical data associated with this genomic information and also with many other things, is really helpful for the research community. But how hard is it, for example, to speak with these stakeholders to increase the infrastructure, for example, in small communities, or maybe to increase the representation? Because we usually see like these big biobanks and really well curated health data sets in big urban areas, but we don't see it in small rural communities. And maybe like I want to get your take on how hard is it for you as an epidemiologist to connect with the stakeholders about making them understand that we really need to improve or quality of data and the amount of data that we're collecting in these biobanks.
Yeah, I think there's a lot to unpack there. It seems like one of the core questions was about urban versus rural. Is that?
Because I feel like to increase the diversity, and so we can, for example, measure how much is the impact of ancestry and ethnicity on different measurements, we need to be more diverse and collect data from usually locations that have like more diversity but are underrepresented in urban centers. So but it's usually like related to cost, because we need to have like a very well established healthcare center to be able to collect this data. But I'm just picturing like you as an epidemiologist and trying to work with genomic epidemiology, you obviously have come in contact with a lot of resistance, and like it's not easy. We don't want to invest in that. We don't see the profit, or we don't see the opportunity. Is it like that, or is improving?
I'm going to call out Victor before he walks away. Talk to him, because he has done some primary data collection. You can go ahead now that you've been called out. Victor has done some primary data collection in Mexico to recruit for a Parkinson's disease GWAS. So I encourage you to talk to him. In terms of geographic diversity, global geographic diversity, there are more and more efforts. And again, you can talk to Paula in the back, because she's done a lot of that in Latin America. Geographic diversity, I can speak a little bit to what's happened in BC, for example, and you're right, we are tied somewhat to big urban centers, because that's where the medical system is. That's where primarily we recruit our participants from. Again, I think that there could be a benefit of COVID, where we can do science at a distance. You can mail a DNA kit out to somebody and then do passive data collection by consenting them. If you get their consent to access their medical records, you can build a biobank that's not based at a big academic institution. So I think that COVID perhaps is helping to break down some of those geographic boundaries in terms of urban, rural within a geographic region, and globally as well. There are efforts, and probably each region has its own challenges to recruit participants and how participants feel about participating in these biobanks.
You have time for one or two more questions? Yep.
Haley?
I was just wondering, so my understanding of GWAS is that you test for SNPs, and those SNPs aren't necessarily like disease-causing variants. But you said that your lab is also involved in all the other omics, like between genotype all the way to phenotype.
Yeah.
So I'm wondering, like, how difficult is it to like determine whether that SNP that you found is like actually causing disease, and like in what way? Like, do you have like a way of doing that?
It's very difficult. It's very difficult. And that's, I think in the early question and answer, we talked a bit about triangulation and multiple different approaches being used to build evidence, because, Will's work, as great as it is, showing colocalization in the placenta in itself is not going to be enough to implicate a single variant or a gene. So it is very challenging. You can approach it from a statistical perspective, and that's mostly what my lab does, is we use different statistical approaches to infer causality. Carl de Boer is another cog in this machinery that uses different approaches to establish causal associations. Wet lab, cell line models, that's all important next steps. We can prioritize those targets from GWAS. Using bioinformatics, we can prioritize targets, but then we need everybody working together, and especially wet lab biologists to functionally validate what we find and establish causal relationships.
Thank you. My question that was actually directly relevant to that. I know of examples where ultimately functional genomics have found causal relationships by SNPs identified through GWAS, and maybe Will can answer this. I'm rusty for MQTLs. Are there any gold standard examples where the specific differential methylation event was shown to be, for example, occluding a transcription factor binding event, but where you have definitive evidence that the differential methylation was basically the bridge between the identified genetics and the environment?
So I will let Will speak. From my perspective, I haven't seen a whole lot of examples where methylation is the definitive bridge. And I know actually a lot of the big MQTL studies show that it's a lot more complex. It's not a linear SNP affecting DNA methylation affecting gene expression. It's a lot more complex how DNA methylation is involved. Will, do you know any definitive examples of methylation? Did you catch the question?
Yes, are you talking about methylation that is causal to disease?
Yeah, I mean, like the last I was looking at was 2017. So anything that's been identified recently where you can, experimentally show that the differential methylation event, the MQTL, was the actual, again, you can go downstream, say there was a transcription factor binding event or something that happened after that. But that MQTL event, that differential methylation, was causal in the studied phenotype.
I mean, I guess I don't have any like super exciting examples. Like an arbitrary example would be when you have a SNP that affects transcription factor binding directly by changing the binding site. And then also that transcription factor binding in and of itself will result in the deposition of methylation around that site. In terms of the methylation itself being causal to disease, there's still ongoing research in like methylation itself actually like affecting the function of transcription factors either like in a very local way or a like transacting like across like several thousands or hundreds of thousands of base pairs kind of way. But tying those to actual disease phenotypes is still very much done like in a high throughput manner, like the research we do. Yeah, I think one of the things that to think about is how can we, as people who study genomics from a like genome-wide perspective, try to make the easiest hypothesis to test possible, and also how do we make sure that that catches the attention of people to do more controlled type experiments. That was, yeah. So we have one concluding question, I think, online that's more about your experience. So if anyone wants to get started with genetic epidemiology, what learning resources do you recommend?
So I would probably speak from my biased perspective of psychiatric traits and being very heavily involved in the genetics of psychiatric traits. And there's this big umbrella organization called the Psychiatric Genomics Consortium. They have or are soon to have online a whole bunch of educational resources that I think really just showcase the coolness of genetic epidemiology applied to mental health traits and phenotypes. So just to kind of get your arms around what genetic epidemiology looks like, I would recommend that. And I think the psychiatric traits have led this move that we need to be doing team science, because so much when GWAS started, everybody was doing their own little GWAS of a couple hundred people. And it was the people who led the psychiatric GWAS who said, we're not going to get anywhere if we just work alone. We need to work together and pool together 20, 30 different GWAS studies to get several 100,000 people in our GWAS, and then we can find something. So I think that's a really cool story that really has been led by the psychiatric genetics community, and they continue to engage in a pretty compelling way with some of the educational resources. So that would be what I would recommend to anybody who's interested.
Okay, well, let's all thank Jess again for a wonderful class. Okay, so that concludes the seminar portion of tonight. We're going to stay here. Jess will be around for the next 30 minutes or so. If you have any additional questions and want to just chat more informally. Okay, so about that.
Yeah, so pizzas outside. I just realized the same place as last time, and as you remember, they cut it like basically 4 slices per pizza, so they're quite big. So I asked each one to take 1 piece first, because there will be enough for everyone. And then if you're hungry, you can go back to the second piece. I'll give everyone have a chance to grab 1 piece. And if you're vegetarian, you can go this way. All the vegetarian pizza and cheese pizza on this side. The meat pizza is on this side. Okay, and you want to grab a piece, and then people can come back in here, or they can stay outside.